Good evening. Welcome to the RP webinar CME program. I'm Dr. Vemuri Murthy, Chairman of the RP webinar CME committee and adjunct faculty, Department of Emergency Medicine, University of Illinois at Chicago College of Medicine, Chicago. COVID-19 has engaged the world physician community in intense efforts to diagnose these cases early and treat aggressively to reduce morbidity and mortality to mitigate the spread of the pandemic. We know the ultimate solution lies in developing a quality vaccine to combat the virus. The speaker of this program is one of the leading authorities on COVID-19 vaccine trials in the United States and internationally. The moderator of the program is Dr. Kalpalata Guntupalli, Professor of Medicine and Chief of Pulmonary Critical Care and Sleep Medicine Section at Bentop Hospital, Baylor College of Medicine, Houston. The 45-minute presentation will be followed by a 15-minute question and answer session. We request you to utilize the chat room for questions to the speaker and complete the pre and post surveys to claim your CME credit. It's now my pleasure to introduce our distinguished speaker, Dr. Peter J. Hotez. This is only a brief introduction to his various accolades and accomplishments. Dr. Hotez is the Dean of the National School of Tropical Medicine and Professor of Pediatrics and Molecular Virology and Microbiology at Baylor College of Medicine. He is also the director of the Texas Children's Center for Vaccine Development and Texas Children's Hospital Endowed Chair of Tropical Pediatrics. He is also University Professor at Baylor University, Fellow in Disease and Poverty at the James A. Baker III Institute for Public Policy, Senior Fellow at the Scowcroft Institute of International Affairs at Texas A&M University, Faculty Fellow with the Hagler Institute for Advanced Studies at Texas A&M University, and Health Policy Scholar in the Baylor Center for Medical Ethics and Health Policy. Dr. Hotez is an internationally recognized physician scientist in neglected tropical diseases and vaccine development. As head of the Texas Children's Center for Vaccine Development, he leads a team and product development partnership for developing new vaccines for various infections, including SARS, MRSA, and SARS-2 coronavirus, among others. In 2006, at the Clinton Global Initiative, he co-founded the Global Network for Neglected Tropical Diseases to provide access to essential medicines for millions of people. He served as the president of the American Society of Tropical Medicine and was the recipient of several prestigious national and international awards, including one from the World Health Organization. I give you Dr. Peter J. Hotez.
Well, thank you very much for that very generous introduction. It's an honor to be able to have the opportunity to speak with you this evening. We've been doing vaccine development for, well, my whole scientific career since my MD and PhD, but for the last 20 years, we've been scaling up production of vaccines for low and middle income countries. I like to say we make the vaccines that the big pharmaceutical companies ordinarily won't make. And I'm happy tonight to share some news with you that we've developed a low-cost COVID-19 vaccine that tomorrow morning will provide an announcement that we're scaling up production in India with a large pharmaceutical company in India. And that's very exciting because there's going, and what's, the Indian pharmaceutical industry has advanced so much that this organization, this vaccine development organization, has the capacity for making 1 billion doses and really could make vaccine for a significant part of the world. And we'll have that announcement tomorrow. So I'll give you kind of a brief hint of what's to come. So I'm going to share my slides, and They said 45 minutes, maybe 45 minutes. I don't think it'll be quite that. Maybe we'll do 30, 35, and we can leave some time for questions. Talking about the COVID-19, our COVID-19 vaccine program, but I want to put it in a much larger context in terms of global health. This is how I want to start. I also like to write books, and this is the latest instant book that just came out by Johns Hopkins University Press, and you can download it for free through their Project Muse. And it's an update of a book I wrote a few years ago called Blue Marble Health, which makes the very interesting observation that when you actually add up where the world's poverty-related diseases are, such as leprosy and Leishmaniasis and Chagas disease and worm infections, tuberculosis. We often always think about the poorest, most devastated countries of sub-Saharan Africa, but on a total numbers basis, they're actually found in the group of 20 nations, the G20 economies, the 20 largest economies in the world. And you might say, well, that doesn't make a lot of sense if they're poverty-related diseases. Well, it's the poor living among the wealthy that now account for most of the world's diseases. And I call it Blue Marble Health to give it a name different from our traditional norms of global health, where we think about developed versus developing. Instead, it makes the observation that all economies are rising, but leaving behind a bottom segment of society. And these are the world's people that really suffer from most of the world's neglected tropical diseases. And I mainly wrote about it with diseases such as leprosy and Leishmaniasis in mind, but in fact, COVID-19 is pretty much going by that same playbook, that the poor living among the wealthy now account for most of the COVID-19 cases. And on the left, I wrote soon, this is moving so fast, we've already exceeded now 20 million cases globally, confirmed cases. And the United States is still #1, but you can see India is really accelerating. India is #3 now in terms of COVID-19 cases, hence the urgency for the vaccine. As I said, you can download this for free right now if you like. And certainly where I am in Texas, we're seeing a very aggressive epidemic with the cases are now starting to plateau and the hospitalizations are going down a little bit, but it's still a very aggressive epidemic here. Even though it's plateaued at 100 miles an hour. Lots of COVID-19 transmission still going on. And again, I like to make the point that it's overwhelmingly, at least in Houston, and I think most of the southern cities where COVID-19 is very aggressive, mostly in low-income neighborhoods. So, now the Houston Health Department every day puts out the list of deaths from COVID-19, and they list them by not by name, but by age and race or ethnicity. And every day it's the same thing: Hispanic, 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 Black, Black, Hispanic, Hispanic, Hispanic. These are the essential workers working on construction sites and family-owned businesses, bakeries, restaurants that are really getting hit. And I think it's a story that we don't tell enough about how it's the low-income people living in low-income neighborhoods that are getting disproportionately affected, very much along the lines of this Blue Marble Health concept. And, and as you might have heard, I've been going a lot on CNN and MSNBC and talking about this and getting more and more upset with the passing weeks. And my wife, Ann, finally said to me, Peter, rather than just giving the weather report every day on how terrible COVID-19, why don't you suggest a solution? So a few weeks ago, I put out in this journal, Microbes and Infection, an October 1 recovery plan, which everybody seems to like except the White House. So unfortunately, it doesn't seem to be advancing. But the idea is, we can get towards containment mode. I think not without a lot of work. In some states, especially up in New England, we're already there. In others, here in Texas, Florida, Georgia, we have a lot of work to do. But the point is then we can do a reset, which is basically what I'm proposing. And then, especially in the South, open up schools and colleges safely. And I still think it's a good idea. It's just been really hard for me to to move the needle with respect to the White House put together that national plan. So we'll see. I still keep hoping. I'm an optimist working on vaccines. You have to be, but we'll continue to push that. Maybe we could talk a little bit about it during the question and answer. In the meantime, we are working very aggressively to accelerate new vaccines. And we think vaccines might be available starting next year. I don't see it happening before then. But one of the things that I do tell audiences is I'm quite optimistic we will have multiple COVID-19 vaccines. And the reason for my optimism is in part because I think it's not that technically difficult to challenge. It's all about making an immune response against the spike protein. So by now, everybody has seen the cartoons of the COVID-19 virus, the SARS coronavirus type 2 that has these spikes that bind to the angiotensin converting enzyme receptors, which itself is an enzyme carboxypeptidase in the membrane of target cells. And if you can induce an immune response, especially ones that include virus neutralizing antibody, you will have a vaccine. And you know, Carl Zimmer, the science writer of the New York Times, says it very poetically. He calls the SARS-2 coronavirus clumsy prey. That's the term he used. And he's right. It's not that difficult to target as vaccine targets go. The problem is figuring out the best way to do it and doing it rapidly so we can accelerate a vaccine. And the problem is often going to take years, sometimes decades, to develop vaccines. How can we compress this safely? And this is the reason why we're having so many different vaccine candidates out there, because we, to really move this quickly, we try to do multiple things in parallel, and we don't really know which is the best way to do this. So for instance, Moderna and Pfizer are both doing this through what's called an mRNA vaccine, which you can make a piece of mRNA very rapidly, gets taken up by the cells, expresses it in protein, and that produces antibody. Inovio is doing this through a DNA vaccine approach. Several companies are doing this through an adenovirus vectored approach. So that's the AstraZeneca, Oxford, and the J&J approach. And then there is an activated virus vaccines. This is being done in China by a company called Sinovac. There's also recombinant protein vaccines, and this is what we're doing in collaboration with this Indian pharmaceutical company. But Sanofi also has a recombinant protein vaccine, as does Novavax, which has a nanoparticle-based vaccine. And then now Merck has entered into this what some people call a race, although I don't like that term very much, through a live virus vaccine, either using a measles platform or a vesicular stomatitis virus platform. And the point is, with with those dozen shots on goal, we can identify which of them might be the most effective at doing this. And this is a slide that Tony Fauci provided to the national academies a few weeks ago. And I like to show it because he included our vaccine that includes the recombinant protein vaccine. But you see there are multiple different technologies. The truth is everyone has advantages and everyone has disadvantages. The nice thing about our recombinant protein vaccine, it uses a well-tested technology, the same technology used to express the hepatitis B vaccine that's produced through microbial fermentation in yeast. The problem is it can take a little longer to produce, although now we've scaled it up. The advantages of the DNA RNA vaccine approaches like the Moderna or the Inovio or the Pfizer approaches, you can make a piece of RNA or DNA pretty rapidly, but it's not a well-tested technology. That technology's never led to a licensed vaccine before. So you can go down the list and we can talk about it at the Q&A. You can basically check the box saying, okay, what are the advantages of this one versus the disadvantages? And we're pushing pretty hard on a recombinant protein vaccine, and I'm excited about it because of its ability to make locally in middle-income countries. And for that purpose, for our vaccine, we've partnered with this very interesting organization that not a lot of people know about, but I think it's really one of the most important global health organizations around. They're known as PATH. They used to stand for the Program for Appropriate Technology in Health. They're based in Seattle. Historically, they've done a lot of work with the Gates Foundation to accelerate a malaria vaccine for Africa and a meningococcal A vaccine for Africa, which they did in collaboration with the Serum Institute of India in Pune. And now they're working with us to accelerate our low-cost COVID-19 vaccine for India. And this is our organization. It's called the Texas Children's Center for Vaccine Development. And our approach is to develop, as I say, low-cost recombinant protein vaccines, established technologies, usually through microbial fermentation. We do other systems as well, but mostly microbial fermentation in bacteria or yeast. And the two ladies with me in the bottom left there, Jill Weatherhead on the right is one of our newest junior faculty members in our center. And to my right, your left, is Mary Elena Bottazzi, and we've been science partners for 20 years, and she's now the co-director with me for the Center for Vaccine Development, advancing a portfolio of the vaccines seen on the right, which are primarily parasitic diseases, which are incredibly important for global health, but they're not financially very remunerative. And as a consequence of that, the big pharmaceutical companies don't get involved. So you might say, okay, well, what does that have to do with coronaviruses? So the truth is we're not really virologists. We're mostly working on vaccines for parasitic diseases. But about a decade ago, we got approached by the New York Blood Center, where their scientists led by Lanying Du and Shibo Jiang had this very important idea that they could develop a recombinant protein vaccine using the spike protein of the coronaviruses for SARS and then MERS, SARS-1, that arose out of Guangdong province in China in 2003. And they had found they could make a great vaccine if they stripped the spike protein down to its barest essentials, just the part called the receptor binding domain that interacts with the receptor. And the reason that's important is because they found that if they used the full-length spike protein or an inactivated virus, they actually got some paradoxical immune enhancement that made it worse. And this was characterized by the presence of eosinophils in the host lung tissue that bore some resemblance to some problems with an RSV vaccine, a respiratory syncytial virus vaccine back in the 1960s. So they had figured out that if they just used the receptor binding domain component, they can make a pretty good vaccine. And they asked us to work with them on SARS-1 and then MERS, and we did. We wrote a grant to the National Institutes of Health, and it got funded, and we've been able to scale up this vaccine. And then when SARS-2 came along, I started getting very worried that this was going to be a problem in the crowded urban areas of low-income countries, because how do you do social distancing in cities that look like this, with crowded urban slums? And I was very worried that these cities are going to get really destroyed with COVID-19. So in Brazil, in Fortaleza, and in Belém, and Manaus, or in Guayaquil, in Ecuador, or in Mexico City, but also in the crowded urban cities, the big cities of India, I was quite worried about it. So we rapidly worked together to start scaling up, at least at the bench level, production of that receptor binding domain, just of the spike protein of the new virus, the COVID-19 virus, SARS coronavirus type 2. And what was really nice, what we were able to show, that all of the years that we invested in making the receptor binding domain for SARS-1 directly applied for SARS-2, and we thought we could move quickly. So we now have developed low-cost COVID-19 vaccines in this yeast called Pichia Pastoris. And you might say, well, who cares about the name of the yeast? And the reason it's important is this type of yeast and related yeast strains are used to produce recombinant hepatitis B vaccine. And countries such as Bangladesh and Brazil and Cuba and India and Indonesia have their ability to make their own hepatitis B vaccine. So it created an opportunity for us to technology transfer our ability to do this. And we started with the original SARS-1 coronavirus with the idea that we can make it as a heterologous vaccine to to prevent COVID-19. But now our team in the lab has worked so quickly that now we've got the SARS-2 receptor binding domain scaled up, and we think we can move pretty quickly on this. So this is what some of the data in the lab looks like. We partner with the Galveston National Lab that's got a transgenic mouse model for coronaviruses using the ACE2 receptor. They also have mouse adapted viruses, and we can get very high levels of virus neutralizing antibody and get complete protection with mice against virus challenge compared to the controls, depending on the different constructs we use. So we're pretty excited about this approach. And this is what immune enhancement looks like. So we keep hearing about the worry about immune enhancement, and some people call it antibody-dependent enhancement, although we think the immune enhancement that actually occurs with coronavirus may not always be antibody mediated, but the point is people talk about it but don't really show what it looks like. So here I'm showing you what it looks like. If you look at the panel labeled E with the group spike protein, you're seeing a bunch of brown dots, and those brown dots are immunohistochemistry staining for eosinophils. So what happens is when you vaccinate and then challenge laboratory animals, you can see this big influx of eosinophils in the lung tissue, in the lung parenchyma. You can also see it in the liver that will cause bronchospasm and other allergic type phenomena. So to avoid this, what we do is we take out the epitopes associated with eosinophilic immune enhancement, and that pairs it down to the receptor binding domain. And now we've done this for both SARS-1 and SARS-2. And again, and we actually find they're pretty similar in structure. Here's the structure of the SARS-1 on the left, in the middle is SARS-2, and on the right, the two receptor binding domains are superimposed. They're highly similar, but different enough that we think the SARS-2 receptor binding domain will be essential. And now what we're doing is we're scaling that up. Now, I'm not going to show a lot of scientific data in this presentation. I'll just try to restrict it to a couple of interesting pieces. And one of them is, you notice in the bottom middle panel there, there's an SDS polyacrylamide gel, which I hope doesn't bring back too many terrible memories of your first year biochemistry class. But if you notice, it looks a little bit smeary and schmutzy. And the reason is because the yeast vector tends to glycosylate heavily proteins. And this is the case with ours. But what our scientists have figured out is if they remove the N-terminal amino acid and asparagine, they preserve the structure and don't have the glycosylation. And that glycosylation can be a problem if you're trying to standardize production. It also affects yield, so you get a nice, crisp, clean band on the right. And now we're able to show that we're getting excellent levels of virus neutralizing antibody using the pseudovirus assay in mice, and now we're doing this in non-human primates. So we're quite excited that we're on a good path to advance this vaccine for the clinic. And tomorrow morning, we're going to announce, hopefully at 6 a.m., a partnership with a large Indian pharmaceutical company who's committed to 1 billion doses of scaling this up. And we'll do clinical testing, of course, but they believe they can ramp up at about $80 million doses a month. The other advantage is the recombinant hepatitis B vaccine is cheap. You can make the recombinant hepatitis B vaccine for $1 or $2 a dose. So we feel that we could have a COVID-19 vaccine for $1, $2 a dose. And if you're talking about vaccinating the world, if you think about it, that's what you need, a proven technology, which you know has a safety record, like had the recombinant hepatitis B vaccine, and it's very inexpensive. And now we're in discussions with Latin American countries trying to figure out a strategy which we can accelerate it there. So these are very exciting times. But as you can imagine, making all this progress, ordinarily taking years, accelerating it in a few months is a daunting task. And so we're waking up early and working long days and into the night to make this happen. Now, the other thing that I'm a bit worried about is as exciting as the progress of science has been in the US, there have been real problems with the anti-vaccine movement in the United States. The anti-vaccine movement really began taking off in 2015, it's a big problem in Texas and many other southern states. And I'll tell you more about that in a minute. But you know, you would think intuitively, with the public clamoring for a COVID-19 vaccine, this anti-vaccine movement would kind of go away or retreat into the corner. And unfortunately, for very strange reasons, it's actually gained strength. And now Reuters and the Associated Press have each done 2 large surveys to find that up to half of Americans will refuse COVID-19 vaccines, even if they're made available. And you're all probably scratching your head saying, how could that possibly be? Well, one of the problems has been the level of messaging coming out of the White House has been very poor. Even calling it Operation Warp Speed gives the impression that these vaccines are accelerated to the point where they're being rushed and not adequately tested for safety. It's not true. We're going through full phase 3 trials for all the vaccines. The warp speed part comes in testing multiple vaccines at the same time, while also manufacturing at risk, meaning you're going to scale up manufacture of this, whether or not you know it actually works, so that when it does, we can move it out pretty quickly. But it still has created a lot of issues and really emboldened this very aggressive anti-vaccine movement. So if you look at the major tenets of the anti-vaccine movement, on the bottom there, one is they claim vaccines cause autism and other chronic illnesses. And I've been fighting with them for many years, and I'll talk to you about a little bit more about that, because one of my youngest daughter has autism and intellectual disabilities, and I wrote a book called Vaccines Did Not Cause Rachel's Autism, about my daughter to fight the anti-vaccine movement. And just as I felt we were kind of winning that battle, then Operation Warp Speed comes along and has played into all the other false tenets of the anti-vaccine movement that claims vaccines are rushed, they're not adequately tested for safety, there's this conspiratorial relationship between the pharma companies and the US government, and this thing that they call health freedom, which means the government can't interfere and tell us what to do. And unfortunately, the way the messaging has happened out of Operation Warp Speed, it's helped play into a lot of those myths, a lot of those false impressions. And now we've got a full-fledged problem in our hands. So what's happened here in Texas is the anti-vaccine movement has basically enlarged its remit. So they're not only protesting against vaccines now, but they're under this banner of health freedom. They're protesting against masks and contact tracing and social distancing. It's really created quite a dilemma, and it's taken on this sort of anti-intellectualism, sort of targeting of scientists, including myself and others. And this is the problem that we face right now in Texas with the anti-vaccine movement. These are the number of kids whose parents opt them out of getting vaccinated. And I'm sure the pediatricians in the audience will empathize with this, up to 65,000 kids being denied access to vaccines in the state of Texas. And a lot of it is based in the Austin, Texas area, where the anti-vaccine movement is quite strong. And now what we're also starting to see are campaigning against this by people holding guns and parading on state capitals. So that's one problem. So I see this fall, the next few weeks, is a time for serious instability in the United States. The other problem that we're having, which is a bit unrelated to the anti-vaccine movement, but it's still quite a problem, is when we did go through that shutdown in March and April, parents did stop bringing their kids into the pediatrician in order to get them vaccinated. And we had this huge drop-off in the number of children receiving their childhood vaccinations, including their measles, mumps, rubella vaccine. That's shown at the panel on the left. It's starting to rebound a bit, but I'm quite worried that now with this sort of neo-anti-vaccine movement, that it won't bounce back fully. So I'm not only worried about COVID-19, I'm worried about starting to see measles epidemics this fall, especially as kids return to school. And then, of course, to make things even more complicated, people are still not getting their flu vaccine. So we have a real risk, I think, of triple epidemics in the United States as we head towards the fall. So I'm very concerned about our nation in the coming weeks up to the election. And then we still have with us this this assertion claiming that vaccines cause autism and other chronic diseases. And and one of the things that they say is they claim the U.S. And Europe have the highest rates of autism because they vaccinate the most. Well, working with the Institute for Health Metrics, what we found is quite the opposite, that actually U.S. and Europe have some of the lowest autism rates internationally. Surprisingly, in India has very high rates of autism, as does the Middle East and parts of North Africa. So these represent some of the higher autism rates. And we're trying to understand the basis for that. And a few years back, because I like to write books, I wrote this book, Vaccines Did Not Cause Rachel's Autism, about my daughter. And it's both a science book, but it's also a personal story. It's published by Johns Hopkins University Press and goes into some detail explaining the scientific evidence showing there's no relationship between vaccines and autism. And the anti-vaccine lobby first claimed it was the MMR vaccine, then they said it was the thimerosal preservative, and then they said it was spacing vaccines too close together, and then it was all about albumin vaccines, and I sort of debunk them one by one. But the other thing I go into is talk about the genetics of autism and all of the genes now we've identified involved in early fetal brain development. So the point is it gives an alternative, explains what actually is responsible for autism. That's me with Rachel there a couple of years back, and kind of goes through papers such as this from Eric Korchesny's group at University of California, San Diego, showing that the patches of disorganization in the neocortex of children actually begin in early fetal brain development. And now the Broad Institute at Harvard MIT has done a really nice study that they published this year in Cell, identifying at least 102 genes involved in the risk of autism spectrum disorder, all but one of them expressed in the cortex of the brain, involved in early fetal brain development. Most are expressed and enriched in early excitatory and inhibitory neuronal lineages, and many of them are involved in the neuronal cytoskeleton. So one of the things that my wife and I did, we did whole exome sequencing of Rachel, and my wife and we've identified indeed a neuronal cytoskeleton gene in Rachel. It's a non-red cell spectrum, a neuronal spectrum. And if you think about it, that makes a lot of sense, in terms of trying to understand the neurobiology and pathophysiology of autism. And as a sort of a digression in the book, but I think an interesting one, we always talk about boys with autism, and we used to say it was 10 to 1 boys to girls. Now we realize it's much higher in girls than we previously realized. Some say 3 to 1 now, male to female, not 10 to 1. And part of it is because autism gets underdiagnosed in girls and women. The girls and women on the autism spectrum often camouflage it better. They're more socially engaged, but they have very high rates of comorbidity, such as obsessive compulsive disorder or even eating disorders. And now we're starting to understand that. And some are saying that this, the sex differences, male, female, may in fact be closer to 1.5 to 1 over time as we get better diagnosing girls and women on the autism spectrum. So I'm spending a lot of time, even though I'm not a neuroscientist and not really an expert in autism, just because of my unique family circumstances, I've gotten kind of pulled into this. But I do a lot of health education around vaccines because I think it's very important in really explaining the risks of vaccines and why they're safe and how they're adequately tested for safety, and trying to counter this very aggressive anti-vaccine lobby that is so dominant now on the internet and on social media. And this is an article that I wrote in the New York Times with Bill Marsh. And if you've ever seen the wonderful Sunday review section of the New York Times on the print edition with all those great graphics, part of that's the genius of Bill Marsh. And I worked with him to model in graphical form what it would look like if you got the disease versus the vaccine. So, and we do this, for instance, on the top left panel is measles versus the measles vaccine, the bottom left, influenza versus the influenza vaccine, bottom right is cervical cancer versus getting the HPV vaccine to prevent cervical cancers and other cancers. So for instance, on the upper left, if in that light colored box is 10,000 people, and this is what happens if you get measles, your likelihood of dying or permanent neurologic injury or deafness, as opposed to your risk of the vaccine on the right, where you can't even really see the dots. We have to kind of draw a line to it for any kind of serious adverse reaction. And some people have found that approach very helpful. One other thing that we're really starting to look at in a very substantive way is not only what vaccines we make, but how we make vaccines. One of the things that I'm very worried about now is we're starting to hear vaccines referred to as the Chinese vaccine or the American vaccine or the British vaccine or the European vaccine. And there's even this new term called vaccinationalism. And I hate it because it really obscures the fact that the success of our ability to vaccinate people against COVID and other diseases is going to depend on international cooperation, like the way we're cooperating now with India. So for instance, on the left, I tell the story of Albert Sabin, who developed the oral polio vaccine from the virus strains in his laboratory at Cincinnati Children's Hospital. But the part not many people know about, in fact, the only reason he could develop the vaccine was he got special permission to bring his virus strains to the Soviet Union, the USSR, and did it right at the time, right after the Sputnik launch in 1957, one of the worst times in the Cold War. The two countries put aside their ideologies to take Dr. Sabin's strains and scale them up into the polio vaccine. And that's where it's first developed and tested on 10 million Soviet school children, shown to be safe and effective. And that's what led to the licensure of the polio vaccine. So two countries putting aside their ideology to work together. And this happened again when the US cooperated with the Soviets on the smallpox vaccine, eradicating smallpox. And it's a story that we've forgotten and we've replaced it with this terrible nationalistic tendencies that I think will interfere with our ability to make COVID-19 vaccines and provide access to the people who need them. So in addition to, and then I'll finish up, in addition to our COVID vaccine program, we're developing vaccines for a number of parasitic infections, including hookworm and schistosomiasis, and our vaccines are now in clinical trials for these diseases. And one of our big problems that we're having is figuring out the market failure. As we move them into phase 3, we really don't have a strategy for how we're going to pay for those big phase 3 pivotal trials, license and distribute them. So it's really a business failure in many ways. So I have many young medical students who come up to me and say, "Hey, Dr. Hotez, I want to go into global health, and I think I'm going to advise them to also get a PhD or study infectious diseases." And they're very disappointed when sometimes I tell them to get an MBA because we need to fix the business model. We need sustainable business solutions for these diseases. And one of them that we're working on now is one very few people know about. It's probably the most common gynecologic condition on the African continent, schistosomiasis, where the eggs, spine-shaped eggs, not only get into the bladder, but they get into the cervix, uterus, and lower genital tract. It causes pain, bleeding, social stigma, and depression, more than 40 million girls and women. And it's linked to a 2 to 3-fold increase in acquiring HIV/AIDS, and we're working on the vaccine for that. And now it's in clinical trials in Uganda and Brazil, and this is the vaccine looks like. It's a surface protein for the parasite that seems to induce protective immunity. We also have a vaccine for Chagas disease, which is a big cause, a poverty-related disease, parasitic infection of the heart. And Catherine Jones, who's leading this effort, has shown how she can block the development of fibrosis with our vaccine, because that seems to be the pathognomonic lesions of Chagas disease, both fibrosis and inflammation. And then this is a disease we're also working on that's a big problem in India, but also in the Middle East, the locals call it Aleppo evil, referring to the city in Syria that's gotten so devastated with the war and conflict there that's interrupted the health systems. And it's very disfiguring for these kids, and especially for girls and women who are often rendered unmarriageable or grounds for spousal abandonment. And we have this very interesting program with the National Institutes of Health with Shad Kamhawi and Jesus Valenzuela, and also the Uniformed Services, where they found out if they add to our parasite recombinant protein, a sandfly protein, which this is a disease transmitted by sandflies, can greatly augment immunity. So we're hoping to move this one in. But again, we kind of get stuck. So we do a lot of exciting work in the lab. We're starting to scale up production. We get to early phase 1, even phase 2 trials, but off with the exception of COVID, because the demand is so high for our other parasitic vaccines, we kind of get held up at the phase 3 stage because raising the money to move it all the way to licensure, since we're not doing this through a pharmaceutical company, with the exceptions of the ones we can identify in places like India that have that ability to scale. So there's a lot of really exciting innovation going on in India right now that we're now tapping into for COVID-19. And we're hoping that we can persuade some of these same organizations to take this on for Leishmaniasis and others. So I'll end here. This is my newest book that I'll have coming out in the first quarter of next year. It's called Preventing the Next Pandemic, Vaccine Diplomacy in a Time of Anti-Science. And to cut right to it, it basically says that we're involved in an epic struggle. And the epic struggle is we are making advancements with developing new vaccines for these diseases, but we're being opposed by an array of 21st century forces. And tonight I've talked about anti-science and poverty, but in the book it goes into more detail about others, such as climate change and urbanization as the world transforms into megacities and political collapse and war. And this is going to be one of our great challenges of how we overcome these 21st century forces. And I always point out that these are not things that we ordinarily teach in medical school or residency training, but maybe it's something that we need to consider. So Dr. Murthy, I'll stop there and see how you'd like to proceed, whether you want to go through some questions or have a discussion. I'm in your hands. So thank you so much for having me tonight.
Thank you, Dr. Hortes, for that wonderful and very thorough vaccines. We have some questions that have come through the chat line here. Dr. Ranga Reddy is asking, how credible is Russia claiming that they have a COVID vaccine?
Well, as I point out, developing a COVID-19 vaccine is actually not that hard. It's an old school problem in virology to induce an immune response against the spike protein. We think a recombinant protein vaccine is the way to do it. Others think an adenovirus. In the case of the Russian vaccine, it is an adenovirus vaccine that they've talked about, uses a combination of Ad5 and Ad26. I'm pretty confident that they've developed a COVID-19 vaccine. What I'm less confident about is whether they've developed one that actually works and is actually safe and has all the adequate quality control and quality assurance. So, and our president makes the same kind of mistake. They think of it as and both Putin and the president and others think of it as a manufacturing issue, that it's some complexity in manufacturing. It's not. It's all about giving it adequate time to test it in large phase 3 clinical trials of 30,000 people to make certain you're not seeing any safety flares or safety signal and confirming that it actually works. And this is what the Russians have avoided. They've sidestepped that. And you see, the way Putin kind of phrases, he calls it, I think he's calling it Sputnik 5 or something along those lines, as though they've made some great technical advance. They haven't. This is not a technologically complex undertaking. What the hard part is it's all about adequate safety testing and quality control and quality assurance. And I don't have confidence that they've done it, given the speed that that's happened. And now what we're also starting to see is sort of threats from the Russians to bypass the WHO, World Health Organization, pre-qualification mechanism, which also ensures quality control, quality assurance. And I'm worried that what they're doing is they're using their influence and the desperation countries are feeling to make one-off bilateral arrangements with African countries and Latin American countries such as Nicaragua or Venezuela or Cuba. And this reminds me of old style Soviet Cold War politics. And that's, again, this business of vaccinationalism. I feel like we're regressing. So I don't find what the Russians are doing is helpful. I'm worried it's counterproductive. And if something goes, and they may get lucky and it may turn out to be okay, but if something goes bad, then it could destroy the reputation of COVID-19 vaccines and the world will refuse to take it. We've seen this before. We saw it in the Philippines with issues around the dengue vaccine, where vaccine programs got interrupted and thousands of kids died of measles unnecessarily in the Philippines because the kids weren't being vaccinated. And you see, even in the Indian newspapers, right, how quickly rumors spread, especially in the tabloids, and it could be very damaging.
Dr. Hotez, there's another question coming from Dr. Veena Govila. She says there's a news that India has almost come out with the vaccine. How are we selecting the patients for the vaccine?
Well, India is going to make actually 3 vaccines at least. So there's Serum Institute of India, which got a great track record, I believe, is mass producing the AstraZeneca Oxford vaccine. I think Bharat is making one there in Hyderabad, and we're working with a pharmaceutical company that we'll announce tomorrow that's making ours. And to vaccinate 1 billion people in India, you're going to need that most likely. And so the question is, who gets it first? And these decisions are, of course, out of my hands. Even in the U.S., how Operation Warp Speed is making those decisions is a little bit unclear. Common sense would say that you want to vaccinate the groups at greatest risk, right? Older individuals, those with underlying comorbidities such as diabetes and heart disease or cancer survivors. But we'll see how India is going to make those decisions for its nation. How we're going to make our decisions is still a bit unclear. In the U.S., we do have good regulatory systems in place between the FDA and CDC, including an organization known as ACIP, for regulating this, and hopefully there won't be any attempt to bypass that.
Dr. Rajiv Arora asks you, which vaccine gives the best T-cell response so far?
So again, one of the things I like to point out is, we're already talking about vaccinating the world, but when you look at the phase 1 trials, they're very limited in scope. So for instance, the AstraZeneca Oxford vaccine that's now being mass produced in India by the Serum Institute of India, it's based on 10 individuals who got 2 doses of the vaccine, because it's only those who got 2 doses that seem to make a pretty good level of virus neutralizing antibody response. Okay, and there are T-cell responses, but I like to give people reality testing. I say, the idea that you're going to now go from 10 individuals in a phase 1 trial to vaccinating the world, there's a disconnect there, there's a piece missing. And it's not just the AstraZeneca vaccine, it's true of the Moderna vaccine. That's 15 people who got 2 doses. With the Pfizer vaccine, it's 12 people who got 2 doses. With the Novavax, it's 56 people who got 2 doses. So we have to have some real reality testing about, how we're going to scale this up, and not being overconfident that these vaccines will necessarily work to the level we want them to and to be safe. Another issue is how well they work. They may be partially protective, and that is they reduce severity of illness, like some of the flu vaccines do in a year where there's not a good match between the vaccine and the flu strain. Or ideally, they would work like a measles vaccine. That's 97% protective after 2 doses against infection. You can induce herd immunity, but we won't know that. So that's a very long-winded way of actually getting to your question, which is, what kind of T-cell responses are there? Well, one of the things that we look for is virus neutralizing antibody, but we also like to see good T cell responses, and most of the vaccines also seem to have pretty good T cell responses, at least in the handful of individuals that we've looked. The Moderna one does not seem to have much in the way of CD8 positive cytotoxic T cell responses, so I don't know if that's going to be a concern or not.
I mean, that's one thing, like at least Moderna vaccine, they're looking at 30,000 people have done 300.
Well, they all will. All of the Operation Warp Speed vaccines will go through 30,000 people phase 3 trials. And that's why we're not going to have a vaccine before the election, as some people assert, because it's going to take a year, I think, to get all of the data on those 30,000 people. And the first one going into phase 3 is Moderna, that's just going into phase 3 now. And you're going to have to look at 2 doses spaced apart. So I don't really see having lots of data on these vaccines till maybe the second quarter of 2021, and rolling out the vaccines in the US to the third quarter of 2021. Now, I tend to be on the more conservative side, and I point out even if we got a third quarter of 2021, that would still be a world land speed record. Dr. Fauci's been saying Q1 or Q2, so you sort of split the difference. So sometime in the middle of 2021, I think, is a likely time.
Dr. Bikasani is asking you, if after vaccine is available, would life be back as before?
Well, it really depends on the characteristics of the vaccine. So again, some of these may just be not just, I don't want to diminish its importance, may work by reducing severity of illness, but may not stop transmission, in which case we're still going to need to have the contact tracing in place and social distancing and masks. So it'll definitely make life better for Americans, give some level for security. But I think we have to get away from this magical solution thinking, which we too often suffer from. And this is the way biomedical interventions are kind of pitched. It's always a miracle solution, whether it's hydroxychloroquine or remdesivir, now vaccines. They will definitely help, but I like to think mostly they're going to be more like companion technologies, not replacement technologies.
Dr. Lola Batu is asking, is this vaccine same like flu vaccine? Do we have to do this every year if a new strain comes out?
I don't think so. I think, there's not enough genetic variation that we've seen so far to require making a new vaccine like flu vaccine. I think these vaccines will, once we develop them and we show that they're safe and effective, now they may require booster shots. That would not surprise me at all. But I don't think it'll be like the flu program where we have to make a brand new vaccine every year.
Dr. Veena Govila is asking, BCG vaccine has any place? I know we have some trials going on at Baylor.
Yeah, there's been a number of people and groups looking at innate immune responses. And even though BCG or MMR vaccine or polio vaccine may not stimulate a protective adaptive immune response, it may stimulate some innate immunity and have some protective effect. I just think by the time my own personal opinion is by the time we figure this out, we'll have the specific vaccines for COVID-19. But I may be proven wrong. It'll be those clinical trials now are underway for BCG, for MMR and polio vaccines to see if they'll stimulate innate immunity in a way that will offer some level of mitigation or protection.
Dr. Mulakutla is asking, are we talking about 60% efficacy or protection with the vaccines? What is the percentage of protection?
Well, we'll have to see. We don't know. We've done a modeling exercise we've done a modeling exercise now with looking at the levels of protective immunity that are going to be required. And it looks like about we're going to need about 70%, 60, 70% protection if the goal is to interrupt herd immunity.
Savda is asking, should I join the vaccine trial? What if I'm in one trial and another company comes out with a better one?
Well, I think that even if you are in a, well, first of all, if you're on a clinical trial, you may be randomized to receive the placebo. You may not, and that's being considered, and then you could still get the vaccine. And even then, it may not prevent you from getting another vaccine down the line. In fact, there may be even some booster effects. So for instance, with our recombinant protein vaccine, the people who've developed some of the adenovirus vectors have looked at prime boost strategies, maybe boosting with the recombinant protein.
They're asking the vaccine that Baylor, you are developing and manufacturing in India, which part of the world would they be supplying and when will it come out? Will it be 1 dose, 2 doses?
Well, I doubt it'll be one. I don't think any of the vaccines so far that I've seen are going to be effective in 1 dose. But 2 minimum, and maybe in some cases 3 doses, and there's a lot of precedent for that. Most of our vaccines, that's the case. And so right now we're scaling it up, production in India, doing clinical trials in India, but where else that vaccine is going to be exported to and used, these are all open for discussion at this point. We're trying to help facilitate that, at least for Latin America.
Please comment on the herd immunity in reference to COVID-19.
There was a question there. I said you can't bounce from one trial to the other. Yes, you're absolutely right. I hope I didn't leave that impression. What I meant was, once you finish the clinical trial, potentially down the road, you'll have the option of getting other vaccines after completion of the clinical trial. I'm sorry I didn't make that clear.
How is herd immunity by cross-infection of coronavirus? And if, let's say, 50% of the population get the vaccine, would there be herd immunity?
Well, there's a lot of controversy about herd immunity. The way herd immunity is often calculated is by using a simple formula of 1 minus 1 over the reproductive number of the virus. So for instance, with measles, the herd immunity is calculated as 1 minus 1 over 12 to 18, which is the reproductive number of the virus. That's how you get to 0.9 or 0.95, meaning 95% of the population needs to be immunized either by the virus or the vaccine in order to get protected. With the COVID-19 virus, the reproductive numbers are ordinarily estimated at 2 to 2.5. And that number would give you 1 minus 1 over 2 to 2.5, and that's how you see that number 60 to 70% commonly quoted. However, others have pointed out that it may not be so simple, that there may be differential susceptibility, so that the actual herd immunity can be quite a bit lower. And the problem is we just haven't seen that yet. If you even look at a place like New York City, which is one of the worst affected areas, the population infected is around maybe 20%. So still far below what we would think of as herd immunity, but we don't know. It may not be that much higher than that. And unfortunately, it's not something you can count on in terms of making a decision about whether you're safe or not.
Can you comment on levemizir as immunomodulator or as a booster?
As levamisole?
Yes.
Yeah, I know.
Yeah.
I know levamisole does have immune potentiating properties, and it may be being looked at now, but to be honest, I was surprised by the question. I haven't seen it talked about in COVID-19. I've seen it talked about in the context of other vaccines, but not COVID-19, but it may be a very viable strategy to look at.
What are the possible side effects of the vaccine? Dr. Jinnah is asking.
Well, again, with 2 doses, there hasn't been a huge, at least in the published literature, there's not a lot. I mean, we only have, 60 and 45 patients in the published phase 1 trials. It looks as though the really high dose of the Moderna one, the 250 microgram dose, did not have a very high tolerability. Same with the 60 microgram Pfizer vaccine, so they didn't go to the second dose. So fever, headache, fatigue, none of them are showstoppers, but this is why we need to do that large 30,000 person study to really look at that in depth. And again, this is why you want to do it, because it may be that the vaccines have modest efficacy, and then you'll have to look at what it really means to have that level of side effects. So I haven't seen anything really terrible that would want me to say, let's not go further. We haven't reached that point yet, so hopefully we won't get there.
Couple of questions on monoclonal antibodies treatment for protection or protection.
Yeah, I'm pretty excited by some of the monoclonal antibody studies I've been reading about. I know, and there's two types of monoclonal antibodies, those that are against the virus, the spike protein, which could be given early in the infection after exposure, and those that reduce the amount of cytokine storm, which also may have a role. The two that seem to bind to the spike protein that I know about are from Regeneron and Lilly in collaboration with AbCellera, and I'm pretty excited about those, provided we can keep the cost down. I always worry that they could be quite expensive. So if we can keep the cost down, I think they'll find quite a lot of use, especially in the time it'll take to develop the vaccines.
Thank you, Dr. Hotez. There are other questions, but I think we'll stop now. It's 8 o'clock.
Okay.
Sudhakar, you want?
Yeah. Thank you, Dr. Hotez, for sharing your groundbreaking COVID-19 vaccine research, along with your inspirational anecdotes. We truly appreciate it. And thank you, Dr. Guntupalli. We are nearing the conclusion of the program, and I request President Dr. Sudhakar Janlagadda to honor our distinguished speaker and propose a vote of thanks.
Good evening. Thank you, Dr. Hotez, for an outstanding talk. And really, it is a very hot topic and sharing your knowledge. Vijay, can you put the slide on, please? On behalf of American Association of Physicians of Indian Origin, recognize Peter J. Hotez, MD, for his outstanding contribution in pediatrics, molecular biology, and microbiology, and welcome him as a member of API Distinguished Speakers Club. Sudhakar Janlagadda, President API, Vemuri S. Murthy, Chairman of Webinar CMA Committee, August 12, 2020. Thank you very much, indeed, and have a good night. Stay safe.
Thank you so much.
This concludes our program. Good night.
Thank you.